Maybe it's time to start. So, welcome to this second lecture in our Forum for Conscious Research. My name is Jan Storm. I'm leading this forum. And as you know, yesterday we had a great lecture by Mark Solms, who presented highly interesting and original ideas about subcortical mechanisms of consciousness. And today, Professor Bjørn Merker will follow up, further challenging the overwhelming consensus that it is our cerebral cortex that is the primary site of our experiences. As Mark highlighted yesterday, Bjørn's work has been a major inspiration for his own ideas. So we're looking very much forward to that. For the benefit of those who didn't attend yesterday's talk, I will repeat some points from introduction then, which unfortunately was a bit hampered by some unexpected technical problems. But both talks are being streamed and recorded and later posted on our website, and you see the address here. And this meeting will last about 2 hours, like yesterday, until 6:30. And after each talk, there will be about 45 minutes for questions from the audience. And since there were many interesting questions yesterday, and more than we could handle then, maybe we can also continue with some of them today, if you like, and time allows. So, as I said yesterday, our field defined the term consciousness simply as experience of any kind, like feeling, seeing, hearing, or thinking, or having emotions, and so on. And this phenomenon is regarded as a deep mystery that doesn't seem to fit into our objective scientific worldview. A key question is where in the brain is consciousness located, and most researchers will answer the cerebral cortex is the main site, but this brainstem arising activation system is necessary to keep the cortex awake. And this answer is also given by Google. If you ask that neuroscientists believe that humans and mammals, the cerebral cortex is the seat of consciousness. And as I mentioned yesterday, among all the 27 prominent scientists who have spoken in our forum for the last 10 years, a large majority here highlighted in red have focused their search for consciousness mainly in the cortex. But prominent exceptions are, in addition to Mark and Bjørn, who was in 2018 already, also Bjørn Merker Sept and Rodolfo Linas. And also, as I also mentioned yesterday, this overwhelming focus on the cortex is also reflected in a recent review article that they published in May this year, where we compare 5 prominent theories of consciousness, but all of them focus on the cortex and not on the deeper, phylogenetically older parts of the brain. But as I mentioned yesterday, I will personally be quite interested in these structures also, although I've never been able to or have the capacity to work on them directly. So when I introduced consciousness as a new topic for medical students about 11 years ago, Bjørn's work on these children with hydroencephaly was an important part of those lectures. So I just showed a couple of slides from there, which you saw some of the pictures yesterday also. And also in talking about hypothalamus, I talk about this sham rage that we also discussed a bit in the dinner yesterday, where cats can apparently show rage even if their forebrain is removed. And this is now this used to be interpreted as unconscious responses, but now this will be reinterpreted in light of Bjørn's and Mark's work. So therefore, it's especially welcome that Mark and Bjørn can discuss so thoroughly these often neglected subcortical mechanisms. So Bjørn Merker is a neuroscientist in retirement, is a long-standing interest in systems neuroscience, evolutionary biology, brain mechanisms of consciousness, and he obtained his PhD from MIT in 1980, and has since then worked on oculomotor physiology in cats, primary visual cortex in monkeys, and song development and mirror self-recognition in gibbons, and the evolutionary development of background of to human music and language. So it's a very wide range of interest, and a couple of years ago we also came with this paper, 2 papers actually, on criticizing the integrated information theory of consciousness. But again, I particularly would like to focus on this, or draw attention to this early paper, 2007, that also Mark mentioned a lot yesterday, about his children with outer cortex, and also the efference cascade is a brilliant paper from 2013, I think, and also work on gamma oscillations, where he points out that they are probably causing activation rather than cognition, and this paper on the IoT, as I mentioned. And these are some earlier papers on interlaminar thalamus and gaze mechanisms, computer-aided neuroanatomy, and female great calls in these apes, the gibbons, and cortex current context and dimensional integration, and also birdsong, music, and language, and so on. So it's a very wide range of interest. I just wanted to mention that, since you are going to only see here a subset. And these are some of the books chapters, in particular the origin of music, with 2 other authors in 2000. So with this, it's a pleasure to give the floor to you, Bjørn, and we look very much forward to your talk. I think your stand's good.
You can hear me. Now you can hear me. All right. And here I see myself on stage. How wonderful. It's a wonderful pleasure to be here. For the second time, this lecture series that you have is a very ambitious project. And I am now proud to be featured with my little picture twice on that. It's really a pleasure with that long list of prominent names in consciousness theory. So thank you for inviting me for this. And it's a hard act to follow Mark's lecture yesterday. I'll do my best, but I can say this, that I couldn't have asked for a better introduction to what I'm going to say than what Mark said yesterday. He has not only understood me thoroughly, but he has himself developed a very profound understanding of the subcortical mechanisms that are involved. At the end of my talk, I may insert a few words about bridging between cortex and the brainstem mechanisms that Mark talked about yesterday. At least I will put up this, a slide of the reference that you just showed, the efference cascade. That was my last word on consciousness. I won't get any more ideas about it than I put in that paper. I'm too old to get new ideas. I'm 81, and because I'm 81, I have written all this out that I want to say, because if I don't follow my script, I go off on wild tangents, and we would spend all night here, and we don't have that time. So, okay, oh yeah, okay, for sure. You put it on me. Okay, thank you. So, without further ado, we humans are the only creatures on Earth that wonder about the fact that we are conscious. Animals just see, hear, feel, and decide without ever stopping to wonder about seeing, hearing, feeling, or deciding. In fact, most humans can't be bothered with such questions either, because nothing depends on the answer. You don't have to figure out how to be conscious. You don't have to do anything to be conscious. You just are conscious. As an automatic background condition for getting on with the real business of life, which is figuring out what to do and when and where. In that respect, consciousness is to the mind what breathing is to the body. Everything depends on it, but we do not notice it unless something goes wrong with it. That is what it means to say that something is a background condition. Other things, the foreground functions, are dependent on it and will not function properly without it. Other things, for consciousness, those dependencies are both many, diverse, and rich. Just think of your capacities and competences while you are unconscious. Given that so much depends on a background function, breathing being a good example, background functions tend to be automatic and to be implemented at basic levels of system function in order to ensure their unfailing operation, their robustness, their smooth problem-free functioning, because so much else depends on them. Now imagine confusing the background function with the functions that are dependent on it. In the case of consciousness, confusing the foreground matters, the contents of consciousness that fills it at any given moment, with that which allows them to be foregrounded, to appear in consciousness at all, namely the condition of being conscious in the first place. Imagine, in other words, conflating the attributes of the dependent functions with the attributes of that on which they are dependent, namely consciousness. That is a deadly mistake, equivalent to confusing the operation of all the things that we can do, given that we breathe, with the operation of breathing itself. An even better analogy is that of a mirror. The properties of mirror reflectance are unrelated to the properties of all the things that may be mirrored in a mirror. Namely, an infinite set of diverse patterns, colors, and shapes without limit, and no amount of scrutiny of the myriad patterns of visible light that may be reflected in a mirror is going to tell you what a mirror is. It would mean trying to derive the properties of the reflector from the properties of the reflected, a deadly mistake. Mirror reflectance is an utterly trivial physical condition, which looks miraculous only because of the richness of all the things that can be reflected in a mirror. But they are not part of the mirror. They are part of the world it reflects. The mirror itself, meanwhile, remains a simple physical object. I submit to you that a deadly mistake equivalent to conflating the properties of mirror with the properties of that which can be reflected in them is being committed on a daily basis in consciousness theory, most generally in the form of assuming that because the contents of consciousness, the foregrounded, are complex and sophisticated, the background function that allows them to be consciously apprehended, that is, consciousness itself, must be complex and sophisticated. So sophisticated, in fact, that nothing short of the final highest fruit of neural evolution on Earth, namely the cortex of our cerebral hemispheres, suffices for its implementation. This typically remains an unspoken, tacit assumption, but occasionally someone spells it out in plain language, as in this opening statement of a chapter on consciousness in a medical text. In man, there seems no reason to doubt that the functional activity of the cortical neurons is essential to the occurrence of consciousness, as the term is usually understood. If any consciousness is possible otherwise, it must be of a character totally different from the phenomenon now recognized physiologically as consciousness. This possibility may be omitted from a physiological discussion. Now, if one interprets consciousness here as contents of consciousness, there is no doubt that in humans, our cortical circuitry contributes and is necessary for much of the material that appears in consciousness. And this may very well be what the author had in mind in making the statement. But the claim the statement makes is not about contents, but about consciousness itself, generically and generally. So who might have said this, and when was it said? I cite it because of how long ago this was said, namely at the beginning of last century. Here is the source. Louisa Burns, 1911. At that time, cortical studies were in their infancy. Brodmann had just completed a decade of labor on cortical cytoarchitectonics with his 1909 monograph on cortical areas. With a few exceptions, such as primary motor cortex, primary visual cortex, and Broca's area, little was known about the cortical function at the time, and very little even of those areas. Louisa Burns' assurance about consciousness being a cortical function can therefore not have been based on evidence regarding the actual workings of the cerebral cortex. It took a good part of another century to work that out. In all likelihood, her assertion, taken as a statement about consciousness itself, the background function, was based on what I just suggested, namely the notion that consciousness is a sophisticated function and therefore must be implemented at the highest levels of our cerebral equipment. Since that is still the dominant consensus among our colleagues today, are we to assume that lots of evidence to support that localization of consciousness itself has accumulated in the meanwhile, making Louisa Burns right in retrospect? In fact, as far as robust and strong empirical evidence for such a localization goes, there is surprisingly little. It consists, strictly speaking, of 3 empirical claims that we shall examine in some detail, one at a time. The first being based on observations on patients who had their corpus callosum severed as a measure to control intractable epilepsy. 1968, 66 and 67, Sperry and Gazzaniga. Severing the connections between the 2 sides of the cortex leaves you with 2 separate consciousnesses, one for each hemisphere. Normally, the two halves of the cortex communicate reciprocally through the corpus callosum, a huge fiber bundle that connects the 2 hemispheres, making it impossible to confine a stimulus to one side of the cortex or the other, because information about it travels across the midline through the corpus callosum. The beauty of that massive fiber connection between left and right hemispheres is that it consists of cortical fibers only, implying that any function that is cleanly split in 2 by cutting the corpus callosum is a cortical function. So this is evidence number 1. Cutting the corpus callosum, if it splits consciousness in 2, the corpus callosum is only cortical, it means somehow cortex is the site of the conscious functioning. Sperry and Gazzaniga claimed that that is exactly what happened to consciousness after the corpus callosum was severed in their patients, namely that it was split in half into 2 parts. And now why would that be? The partial decussation of the optic tract at the chiasm ensures that information from each visual half field reaches only the opposite hemisphere. Okay? Not each eye. It's not that the left eye reaches the right brain and the right eye reaches the left brain. The whole visual half field for both eyes, left half of this, what the left eye sees and the left half of what the right eye sees, reach the right hemisphere. And the right part of each half field reaches the left hemisphere. So, by presenting stimuli to the right or left of visual fixation, which lands the stimulus in one half field or the other, you can make sure that it travels up to the opposite hemisphere. So if you present the stimuli to the right or the left of visual fixation, you know it ends up in the cortex on the opposite side. And in the patients with a cut corpus callosum, you can be sure it stays on that side because the cortical communication across the midline has been cut. Now, each half of cortex controls the hand on the opposite side of the body. But language is typically left lateralized, so only the left hemisphere speaks and talks, as it were. If, therefore, one asks a split cortex patient to respond both verbally and manually, they should be able to respond both verbally and with their right hand when the stimulus is presented to the right of fixation, which sends it to the left hemisphere, which talks, so they can say, yes, there it is, and they can palpate with their right hand, which goes also to the same left hemisphere. They can palpate something and say, that matches what I saw. If, therefore, one asks a split cortex patient to respond both verbally and manually, they should be able to respond both verbally and with their right hand when the stimulus is presented to the right of fixation, which sends the stimulus to the left talking cortex. But when presented to the left of fixation, it sends it to the essentially mute right hemisphere of the cortex, and they should be unable to report verbally about the stimulus, but should still be capable of responding with their left hand, which the right side controls. Okay, left side of fixation, left hand, but no talking. According to Sperry and Gazzaniga, patient performance conformed to this pattern of anatomical expectations. Not only had their visual world, as expected, been split in half, it's inevitable, but the agency involved in responding to the stimuli also seemed split between the two halves of the cortex. It seemed, therefore, that the full gamut of conscious functioning that was engaged in their testing, perceptual awareness as well as agency, had been split in two by the cut, from which they concluded that two separate consciousnesses were present in these patients, one for each half of the cortex. This claim, then, is our first piece of empirical evidence for a cortical localization of consciousness, and we shall return to it after looking at additional evidence pointing in the same direction. A year after Sperry published his claim about two consciousnesses in split-brain patients, Humphrey and Weisskranz reported behavioral observations in macaque monkeys with complete bilateral removal of the striate cortex, that is the primary visual cortex. What you see on the slide there is visual cortex removal eliminates phenomenal vision while leaving visually guided reaching intact blind sight. That statement is not in the 1967 paper because it was on monkeys, and Humphrey and Weisskranz didn't have access to the consciousness of monkeys. We don't have that, only their behavior. But as soon as this was followed up in humans, where we have evidence through verbal report of consciousness or not, this was confirmed. So the monkeys in their paper were considered blind, but they didn't know that they didn't see anything. But they coined the term blind sight. They were working in a venerable tradition going back to Heinrich Kliever and more generally all the way back to Florence in the 1800s of assessing deficits and residual capacities after brain damage. At the time, the removal of striate cortex in macaques was thought to devastate their capacity for visually guided behavior, making them essentially blind, leaving only rudimentary discrimination ability of crude parameters such as total luminous flux. And note that we are talking only about behavioral measures here and not visual experience or consciousness, which is not accessible in the macaque monkeys. By carefully training and testing their animals, who had already been through extensive testing on a variety of discrimination procedures, Humphrey and Weisskranz were able to show that their visually guided behavior extended far beyond what had been reported so far and included the capacity to localize and reach for visual stimuli in their surrounding space, including stationary ones. This was sufficiently noteworthy to be published in Nature and stimulated studies to identify the visual competence of humans with striate cortex damage. Now, in human patients, unlike the monkeys, the damage to visual cortex is typically incomplete, but it leaves a so-called scotoma, a region of the visual field corresponding to the damage in which the patients, for all practical purposes, are blind. If one controls their visual fixation behavior, patients can therefore be assessed for residual visual capacities in the blind part of the field. They will typically protest that they see nothing in that part of the field, but with repeated coaxing and encouragement and the instruction to simply guess, they prove capable of reaching for and grasping a variety of stimuli presented in their blind fields. And now, with human patients rather than monkeys, we have access to what the patients experience, to their consciousness, to phenomenal vision, not just the behavioral responses in the monkeys, through their verbal reports. And these patients typically claim not to see the stimulus they successfully reach for. The most tested of these patients, and he accounts for the overwhelming number of published reports on blind sight. This most tested patient, who was tested in labs all over the world, he became a kind of blind sight jet setter, this man. And he goes by the initials GY. He was adamant that his reaching was not guided by any visual impression at all. He said he experienced something, but it is not visual. He was very emphatic about that. He was a claim, in other words, that successful reaching for visual stimuli was unaccompanied by any visual phenomenal experience. And this was termed blind sight, that is, the paradox of visually guided behavior in the absence of visual experience. And this, of course, makes blind sight highly relevant to the localization of consciousness, because it implies that visual cortex is necessary for visual experience, for phenomenal visual experience, because removal, loss, or damage to a piece of visual cortex abolishes visual experience, visual consciousness, in that part of the field of vision. So this is our second piece of empirical support for the stance that consciousness is a specifically cortical function. And here comes a third piece of convergent evidence for the same stance. For a stimulus to reach consciousness, its cortical feed-forward and feedback activity must converge. Already in the late 1970s, Gerald Edelman proposed that interaction between feed-forward and feedback activity is involved in consciousness. He called it re-entrant signaling, and typically attributed it to the thalamocortical complex as a whole. That is, his conjecture was not necessarily confined to the cortical circuitry itself, but it included it. It was Victor Lame who formulated the idea in terms of cortical circuitry specifically, and worked out experimental procedures using masking and priming stimuli by which he followed and manipulated stimulus progress up the cortical hierarchy with close to millisecond precision. Lame's conclusion, based on a line of studies spanning more than 2 decades, is that the stimulus enters consciousness only when its feed-forward cortical activity meets with returning feedback activity evoked by the same stimulus in the bidirectional circuitry of the cerebral cortex. Now, if that is the case, we have yet another piece of convergent evidence that ties consciousness to specifically cortical activity, and this time not only with respect to the overall functional properties of the system, but tied down to the very circuitry by which the cortex operates. We will take a closer look at the matter of cortical organization later, but for now, to summarize, here are 3 key references as access points to these independent but convergent lines of support for a corticocentric view of brain localization of consciousness. 1 key reference for each of the 3 pillars of support I have just reviewed: split brains, blind sight, and feed-forward feedback convergence, Victor Lame. So those are the three key references. Good access points. In other words, if you're not familiar with the literature, you can start with those 3. There are, of course, additional findings claimed to support a corticocentric view, from the effects of sleep and anesthesia to various physiological results related to ignition, broadcasting, and the like. As far as I can tell, they do not, however, make quite as clean and compelling a case for a cortical locus as do the 3 we have just considered. For example, some of the anesthesia evidence points to a subcortical primary locus of difference between consciousness and anesthesia. And for ignition, there are subcortical sites that correlate better with stimulus awareness than do the cortical sites where ignition was first studied. So all in all, I think the best we can do for the corticocentric position are the three principal lines of empirical support I have just highlighted. The fact that they provide quite different kinds of support for the corticocentric stance only enhances the strength of that support. And taken together, those three would certainly convince me that consciousness is a cortical function. That is, that the cerebral cortex is the organ of consciousness. But since I am on record for opposing that position, presumably I am not convinced that these claims are quite what they have made out to be. And as it turns out, these claims are not only not quite what they seem, they turn out to be wrong. As follows. Starting with Sperry and Gazzaniga's claim regarding the split brain, careful reading of their reports shows that as far as the agency part of their observations go, the evidence was not quite as unequivocal as their conclusion would demand. They were also not based on quantitative assessment of patient behavior, but more informal clinical testing. These weaknesses induced a group of researchers that include Michael Corballis, an old hand at cortical function, and Victor Lamme, to review the entire question of the unity of consciousness in split brain patients and to present new quantitative data of their own regarding patient behavior on the same very tests that Sperry and Gazzaniga performed half a century ago. Here is the reference to one of the group's major reports from 2017. Split brain patients are hard to come by these days. This operation is no longer performed. But these researchers were able to find 2 of them and get their cooperation for an extensive series of quantitative tests that extended over years. The behavior of these patients contradict the split consciousness interpretation of the original, by now classical, reports by Sperry and Gazzaniga. Reports that are featured in every textbook on neuropsychology and neuroscience, and incidentally helped Roger Sperry win the 1981 Nobel Prize in Medicine. I can do no better than to let the authors of the 2017 report summarize their findings themselves. Oops, what happened here? Okay, I'll have, oh, it didn't forward. Oh, pushed the wrong button, that's why. Okay, so there we go. That's agency for you. Across a wide variety of tasks, split brain patients with a complete and radiologically confirmed transection of the corpus callosum showed full awareness of presence and well above chance-level recognition of location, orientation, and identity of stimuli throughout the entire visual field, irrespective of response type, left hand, right hand, or verbally. Crucially, we used confidence ratings to assess conscious awareness. This revealed that also on high-confidence trials, indicative of conscious perception, response type did not affect performance. These findings suggest that severing the cortical connections between hemispheres splits visual perception but does not create 2 independent conscious perceivers within one brain. Now this, of course, is a severe blow to the split brain support for the corticocentric stance on consciousness, a piece of evidence that has essentially vanished with this re-examination of the matter. Knowing exactly what these findings imply regarding neural mechanisms of consciousness and their localization involves additional subtle issues that have been addressed by these same researchers and additional colleagues. And here is the reference to their follow-up paper a few years later, 2020, where they discuss what these findings imply and what we still have to find out to be sure about localization. But as far as the original classical story goes, we can simply cross it out from our listing of the 3 pillars of the corticocentric stance. Like that. Sperry and Gazzaniga are gone. So one pillar has fallen, and we go on to the next one, blind sight. The phenomenon called blind sight generated a whole industry of studies and controversies after its original discovery, stimulated by the Humphrey and Weisskran observations on macaques. Generally, one simply took patients' statements that they did not see anything in the affected part of their visual fields at face value. Verbal report is sometimes called the gold standard for consciousness assessment. They were claiming all, we don't see anything. They were blind in the scotoma, part of the field. No phenomenal vision. It took years till someone decided to find out what the something that was not visual that G.Y. claimed to experience inside his scotoma in relation to the stimuli he reached for, what that something really was. Perhaps it was after all visual, but of a low-level degraded kind that the patient would not call visual because of its stark difference from the rich world of shaped, colored, and textured three-dimensional objects plainly visible to the patient in the good part of his field. To make a long story short, one of the principals of the blind sight genre, Petra Störig, teamed up with Erhard Barth, whose skilled programming generated a world of degraded visual stimuli, and they proceeded to ask G.Y. to match the something he experienced in his scotoma to those computer-generated stimuli shown in his good visual field. So here is his good part of the field, here is his scotoma, here is the something that he claims is accompanying his reaching behavior, but it's not visual. And so here we have the stimuli that they generated artificially, one after another, a huge corpus of stimuli they had generated. For almost every one of the large corpus of artificial degraded stimuli shown to G.Y., his answer was, no, that's visual. Till they hit on a couple of contrastless, contourless visual stimuli that were defined by only a difference in texture grain or noise statistics from background. And G.Y. said, yes, that matches the something well. So that was that. G.Y.'s something was after all visual and phenomenal, despite his insistence to the contrary. And so the principal patient of blind sight studies had after all phenomenal visual experience inside his visual cortex scotoma. And here is the reference to the Störig and Barth report. I have highlighted in red, phenomenal is in the title of the paper. Phenomenal visual vision despite unilateral destruction of primary visual cortex. Störig and Barth are not the only ones who have reported such results from patients. They have shown low-level phenomenal experience, imputative blind sight, Semir Zeki among them, and Axel Kleremans. So blind sight is no longer blind. And with that, the second pillar of the corticocentric stance on the localization of consciousness falls. Well, it is obvious by now where this presentation is heading. But to topple the last pillar of the corticocentric stance will require going into a bit of the intricacies of cortical connectivity. First of all, the evidence Lamme has marshaled that convergence of cortical feed-forward and feedback activity is necessary for a cortical stimulus to reach consciousness is convincing and has stood the test of time, and I take it to be an empirical given. In other words, feed-forward feedback convergence in cortex for a cortical stimulus is necessary for it to enter consciousness. I take that as a fact. This is not going to be challenged by what I say here. However, for that empirical finding to be evidence that consciousness is a cortical affair requires more than the fact that cortical feed-forward feedback convergence is necessary for cortical activity to enter consciousness. It requires the further step of showing that it actually is in the cortex itself that this convergent activity becomes conscious and not somewhere else. It turns out that not only are there good grounds for believing that exactly that cortical activity, which is subject to feed-forward feedback convergence, is shipped out of the cortex to subcortical target loci, but it has also been shown empirically that it is by thus being exported subcortically that that activity becomes conscious. Let me try to explicate this by means of a diagram. And here we're going to have to enter some of the intricacies of cortical circuitry. I spent days preparing this slide for this session to guide you through the jungle of cortical connectivity. I got so engrossed in making this slide. I liked the way it looked so much that I got carried away on the left part of the thing, which is the layer 6 connectivity, which is not relevant to what I'm going to talk about, that I spent so many days on it that I never got to what I wanted to do, which is the right part. In the right part, I only have the cortical neurons, so I will fill in the... How can I point? Is this a pointer?
Use the mouse. You can use the mouse.
The mouse also, okay. Okay, so oh, that was a high gain on this mouse. So here is the higher order thalamus. This is the first order thalamus. I've divided the two thalami in half. This is not left and right side. This is simply the nuclei that belong to the left, to the first order thalamus, which is the standard ones: lateral geniculate, medial geniculate, ventrobasal complex, primary sensory information going up through the thalamus to cortex. That has very little to do with consciousness, except for the fact that the stimulus has to get to cortex. The higher order thalamus is the one that is relevant to this, and I will fill in that part with another hastily assembled slide when I realize that I won't have time to finish this. But this one will do fine for orienting ourselves to the cortical part. From outside and in, layer 1, layers 2 and 3, which most people who do with these studies lump together. They can be divided up into 2 and 3, but they are very similar, those cells. Layer 4, which is a primary recipient layer, which does not project long distance across cortex, but only to other layers. Then layer 5, which projects long distance across cortex, and layer 6, which does project long distance across cortex. And here are these pyramidal cells. A sample of them, here is if you take the left and right side together, it's a full sample of typical cell types of the pyramidal cells, not the interneurons. They are small and not part of this picture. The reason they look so knotty, so uneven, is that I have tried to emphasize the fact that the surface of cortical pyramidal cells is covered with dendritic spines. There's recipient little protrusions, little like mushrooms that stick out from the dendritic surface, and they receive the synapses from the afferents. And the cortical cells are studied with thousands, literally thousands of these spines. So to remind us of that, I have made them knotty. uneven, the surface. These are all the little spines that I symbolize with that. And because the dendritic surface with the spines that receive the afferents are the receptive surface of cortical pyramidal cells, where they receive their information, where they extend their dendrites, their tentacles, tells us what they are interested in, what those cells want to monitor, what those cells are integrating. Okay? So here you have a layer 4 cell, a stellate cell in layer 4 is not interested in anything else except stuff happening in layer 4, because it doesn't extend its dendrite into any other layer. There's a neighboring cell in layer 4, also a layer 4 cell. It does have some interest in other layers. It sends its apical dendrite up into layer 1, which is a very peculiar layer, which we will get to. Here are other cells. Here are cells in layer 6. This one is only interested in stuff happening in layer 6. And look at it. It's upside down. The cell body is on top and the apical dendrite is on bottom. The cell is upside down by regular pyramidal cell standards, but layer 6 is a very peculiar layer. It has cell types that nobody has ever seen in any other layer. It's a very peculiar layer, and it's very important for the consciousness story, because it regulates global state. It regulates the whole spectrum from delta up to high gamma. It correlates with activity in layer 6. It's recent findings. It's a profoundly important layer. Matthew Larkham is involved in unraveling layer 6 now. He's in full blast studies. He has a couple of papers out on it already. It's a fascinating story. The other layer that is relevant to consciousness is layer 1, where context and attentional factors are integrated in a long-range global way. So layers 1 and layer 6 are crucial layers for knowing the cortical part of cortical involvement in consciousness. So here in layer 6, we have one that is interested only in layer 6. This neighbor of it extends its apical dendrite up into layer 4. It wants to know something about the primary afferents. And this one, also layer 6, sends it all the way up to layer 1. So here's layer 6 communicating with layer 1, the two ones I said are important for regulation of consciousness. But let's return now to layer 5, because that's what this story is about. Look at this beast. It's interested in activity in every layer of cortex. The cell body is down in layer 5. The cell body is actually down in layer 5b, which is where the big outside of cortex projecting pyramidal cells are located. There are also pyramidal cells in layer 5a. I haven't shown them here. Now, what the dendritic distribution of this cell tells us is that it's interested in what happens in layer 6. It's interested in what happens in layer 5. It's interested in what happens in layer 4. It's interested in what happens in layer 2, 3, and in layer 1, it's very interested. Just by looking at the dendritic distribution, you can tell what that cell integrates. Not only does it integrate across all layers, that is, it is presenting its cell body. The spiking at its cell body is an integrated summary of everything that happens in cortex. It's a cross-section across all the layers. Who might be interested in that? Somebody else outside of cortex who doesn't know what's happening there might be interested in that. And we'll come to that. But look at this thing, this branching dendritic apical dendrite that extends through layers 2, 3 and up into layer 1. Matthew Larkham, with his exquisite physiological studies, has shown, his lab, he has many collaborators, but he is the principal investigator there. He has shown that there is special conductance mechanism in this apical dendrite of layer 5 pyramidal cells that amplify the afferents taken from layer 1 and layer 2 and 3, but primarily from layer 1. and therefore that this apical dendrite, which is far from the cell body and would normally have a minimal influence on the cell body and the spiking that this cell actually sends out to the rest of the brain, that amplifying special conductance mechanism, which is calcium spiking, moves this down so that it has a profound influence on the spiking of the cell body. So now you have a special amplifying mechanism to pick up, to make extra sure that everything that this cell is picking up is shipped out into its axon. This cell, in other words, is ideally situated to integrate feed-forward and feedback activity, just as Victor Lamme says, and just as Edelman suggested is essential for consciousness. And it does. Other studies from the Larkham lab show that feedback activity, a prominent stream of cortical feedback activity, travels in layer 2, 3, and 1. And the calcium activity in the apical dendrite of these layer 5 pyramidal cells correlates with a fluctuating feedback coming back across cortex in layer 2, 3, and also 1. Okay? So now we are building a case here that this beast, this huge cell, which are the biggest cells in cortex, the layer 5B pyramidal cells, are the big ones. That this one may be a crucial part of the integrating of feed-forward and feedback activity that Lamme claims is crucial for consciousness. Where then does this activity that might be critical for consciousness end up? Well, unfortunately, layer 5B pyramidal cells, on the cortical localization question, we would like to know, does that activity that is integrated by this cell, does it stay in cortex and become conscious there, or is it shipped out and becomes conscious someplace else? We don't know, because these layer 5 pyramidal cells project profusely across cortex, and they project profusely downstream into subcortical structures, not just close to them in the higher order thalamus, but all the way down to the colliculus and all the way down to pons and medulla, all the structures that Mark Solms talked about yesterday. They project downstream in a big streaming axons with branching to the different reticular structures in the brainstem. Okay? So here is a candidate. What if we could find out, what if we could separate the stuff that these cells send across cortex to other cortical areas, or if we could separate that from the stuff it sends downstream? Well, a miracle has happened, dear friends. A miracle has happened. The Larkham lab has, by using the latest genetic and optogenetic methods, have managed to separate the output from layer 5B pyramidal cells, which are very likely crucial ingredients of what makes a cortical stimulus conscious. They have managed to separate them into transcortical output and downstream subcortical output. How have they done that? We are so lucky that the layer 5B pyramidal cells that send their axons across cortex are one cell type, and they are separate from the cell types that send them down subcortically. And those two cell types have molecular markers, which means that you can use the modern channel rhodopsin and so on, optogenetics, to selectively record from and manipulate the transcortical and the subcortical output from layer 5B. And what they found was clear-cut evidence, reported in Nature Neuroscience 2020, that what correlates, this was done in live mice, sitting with the optogenetic headpiece, and it's only the what correlates with stimulus awareness in these mice. This was whisker manipulation in the barrel field of somatosensory cortex. It's only the downstream activity of these cells that correlates with stimulus awareness. Okay? Now, you don't know whether the mouse is aware. We don't have access to its phenomenal experience, but the mice who were fully awake, they were... Oh, I forgot to tell you. How do they manipulate it? Optogenetically and pharmacologically, they can selectively turn off the layer 5B cortical synapses without turning off the downstream synapses, and they can turn off the downstream synapses to higher order thalamus, colliculus, and Mark Solm's brainstem without turning off the cortical one. And the outcome was completely black and white. It's the transcortical cells do not correlate with stimulus awareness. The animal was trained to lick when it saw the stimulus, and they did psychophysics on this. They did the full receiver operating characteristic. They did the whole thing. Here is the reference. Oh, sorry. Here is the reference. And backwards, I guess, is going here. So here we have this one. This is where I complete the picture. Here's the mouse. Here is one of these beasts. I've compressed it a little. Here's layer 1, layer 5B. And it sends its axons transcortically, and it sends them downstream to the higher order thalamus and to the colliculus, and further downstream to the brainstem. When they turned off these transcortical fibers, turned off all the synapses, performance and evidence for stimulus detection was unaffected. When they turned these off, performance fell to random. Okay? The mice showed no... They could actually run the threshold for detection up and down by manipulating the excitability of this part of the apical dendrite. The amplifying part of the apical dendrite. They could themselves decide to the threshold of the mouse by running excitability selectively only for these neurons because of the molecular coding. This is very strong evidence, not only that they are correlated, but that they are causally involved in launching that stimulus to awareness. And for that stimulus to reach awareness, it has to be shipped subcortically. With that, of course, we have a piece of evidence that the Lamme convergence of feed-forward and feedback is indeed a condition for a stimulus to enter awareness, but that awareness, in all likelihood, takes place subcortically, somehow. So, here comes the reference again, which you saw. And so, back to our 3 pillars. Edelman-Lamme, for a stimulus to reach consciousness, its cortical feed-forward and feedback activity must converge. Yes. But it doesn't mean that it becomes conscious at the cortical level. Indeed, we now have this Takahashi and Larkham evidence that it is by being shipped subcortically, by being exported to subcortex, that it becomes conscious. And so with that, we can cross out the 3rd pillar of corticocentric evidence. So what are we left with? As far as robust and specific evidence goes, the answer is, strictly speaking, nothing. Now, of course, what I have done now is taken what I consider the strongest evidence for a corticocentric, the strongest empirical evidence for a corticocentric stance on the localization of consciousness, and I have said that that evidence, as originally reported, does not hold up. There is strong empirical evidence against each one of the 3 pillars. That doesn't mean that cortex has no role in consciousness. Like I said at the very outset, obviously the stimulus information that is generated by cortex, such as three-dimensional objects, is dependent on the cortex, no doubt about that, the contents. But if you want cortical content to enter consciousness, you have to have a cortex. But where it's located, the localization of the consciousness mechanism becomes now a separate issue. And see. Now it becomes possible to understand previous very puzzling findings. And here is one of them. A standard test in perceptual psychology is visual search tasks. You're presented with a screen, and there's a lot of capital T's scattered across the screen, and among those capital T letters is a capital L. One capital L and 30 capital T's. And you sit in front of the screen, and this is all done, of course, in very precisely timed stimulus presentation. Your task is to find the L among all the T's. And it's quite a challenging task, actually, when you do that. If you have been a subject for such experiments. It's called a visual search task. The human subject is obviously instructed. Your task now is to find the L among all the T's. Go. The instruction obviously ends up in frontal cortex. This is an instruction in language. If it's an animal, you have to train the animal because you can't instruct it verbally. You have to train it, so there's an elaborate conditioning procedure to get it to follow the instructions, to find the L among the T's. Okay? But you can do that with animals. It's done routinely in animal experiments. And of course, that routine training to get it to understand what the experimental task is also a frontal thing. So here I have put a highlight in frontal cortex, which is simply a symbol for the frontal instructions, the executive instructions that the animal uses, or the human uses. Oh, by the way, this is how real brains look. This is from Valverde's Golgi and a 2.5-day-old mouse. Golgi stained, and he was lucky. You know, he made a whole atlas of this brain. Lots of things stained in Golgi. Often it's just a few neurons that stain. Here the whole thing stained. It's beautiful. So here I have put on Valverde's diagram, or actual Golgi picture, I put the frontal, prefrontal executive instructions to find the L among the T's. If you had asked me before the study I will show you, if you had asked me 14 years ago, how does the brain ensure the biasing of perceptual apparatus, which is posterior cortex, visual cortex and somatosensory, and hearing is back here, how does the frontal cortex instruct the sensory mechanisms about what the task is? Well, I would have said, obviously, it uses the feedback connections from top down. It uses the cortical feedback connections, which we know are there. It uses them to instruct the sensory areas about what is wanted, namely an L among the T's. That's what I would have sworn, and there's no reason to think otherwise. We know the feedback connections, the top-down connections are there. Why not use them for this task? Frontal cortex executive instructions, and bias the sensory areas for L's versus T's. That's not how it's done. So not how it's done. Rather, Lovejoy and Krauslitz, 2010, had this set up with monkeys, trained monkeys, experts at finding the target stimulus among distractors, well-trained, over 80% criterion performance, and they happen to have a micro-injection cannula in the superior colliculus. Now, if it's feedback connections, top-down connections from frontal cortex to posterior sensory cortex, the colliculus should have nothing to do with this. The instructions, they should still be able to follow the instructions. They injected micro-injections of muskimol, which paralyzes, which deadens, it inhibits, actually, the neurons in the colliculus. They made micro-injections, small local injections, corresponding to the site where the target was on the screen, okay? So it's a very selective, very precise study. 2010, Lovejoy and Krauslis. Lo and behold, as soon as they made their micro-injection, performance was devastated. Evidently what happens is the frontal instructions go all the way down to the colliculus, intermediate and deep colliculus, which frontal cortex projects to. From there they go up to the pulvinar, and with one synapse in the pulvinar, the instructions go up to the sensory areas. Why not use the short route? Well, that short route isn't as short as you think. Many of the top-down connections are relays of multiple projections from area to area. Some of them are long, but not all that many. So that may take as many synapses to get to the sensory areas from frontal cortex by top-down connections, by feedback connections, as this swift 1, 2, boom, up. Okay? This is how it's done. It's surprising. I would never have guessed it, but that's how it's done. And it has to do with what the colliculus is. The colliculus is a priority gate. It sets the priorities for the very next orienting movement. Okay? Now, oh yes, let me finish this by saying the quote from the Takahashi et al. From the Larkham lab in their big report in Nature Neuroscience 2020. At the end of the discussion, they have the following sentence. They are the ones, that's the paper where they showed that what's critical for the stimulus detection, for the threshold of stimulus detection, for the stimulus to reach awareness, is layer 5B pyramidal cells and their downstream subcortical projections. Where do they go? They go to pulvinar, they go to colliculus, they go to the brainstem, and they ask the following question. They make the following statement. It remains to be determined why sending this information to these specific subcortical regions, POM and superior colliculus, is decisive for perception. Well, my answer is it's decisive for perception because the crucial mechanism of consciousness is subcortical. It has to get there to become conscious. That's my answer, and I'm very happy to report to you that I made this prediction 7 years before this result was reported. I made it in paper where I cited previous Larkham studies about the apical dendrites and the amplification and their relevance to attention and context and all of these things. I cited those Larkham studies, and I stuck my neck out, and I said, I predict that the consciousness mechanism is a complex of pulvinar, higher order thalamus, and colliculus working in tandem. It's a very complex paper, and I will leave you the reference here. Oops. Oh, it's not there. Okay, it's the slide. It's the reference. It's called the efference cascade. It's a frontier paper, so it's easy to get access to it. It's called, if you just Google the efference cascade, you get the paper. The efference cascade. You had it. I didn't include it in the ones I sent you. I included them in my own. Okay, so it feels rather rewarding to be confirmed in a 7-year-old prediction this way by Larkham and colleagues. And I met Larkham for the first time just like I met Mark Soames for the first time in Kathmandu, Nepal. In May of this year, there was a wonderful conference on consciousness in Kathmandu, Nepal, following up on the one in Dharamshala, India last year. And there I met Matthew Larkham. You may have noticed that it said nothing about consciousness in his title. He is the most humble man you can imagine. It said perception. He is not trumpeting his findings. He is a very humble and unassuming man, and so you wouldn't know it from the title of his paper. I noticed it instantly, of course. I hit the ceiling when I saw, oh my God, I proved what I said 7 years ago. So thank you for that. That's my take on the corticocentric evidence. And I have left plenty of time, I hope, for questions. Well, look at that. Less than 1 hour. Yeah, very good.
Thank you very much, Bjørn, for a wonderful talk. It was really marvelous. And these two talks together, I think, is the best I ever had in our forum.